Good afternoon, everybody. As Bill said, I'm Rachel Christensen. I'm an undergraduate researcher here at the University of Utah and working in the Natural History Museum with Dr. Power and Dr. Gray. And my project this past year has really been to understand how there's stomatal changes and responses in bristlecone pine to changing atmospheric conditions. So it's an interesting story, and we're going to dig into it. So just to start with the framework of this study, where I began a year ago, was simply using stomatal density to track changes in CO2 in parts per million. So this figure here from Van de Water is using Pinus flexus, so a different variety. It's limber pine from pack rat middens in the Great Basin Desert. And the main takeaway from this paper was that we can see an inverse relationship in stomatal density and atmospheric CO2. So that big crux of it right there. And this is through geologic time, through interglacial and glacial cycles. So we're seeing very different flora in those two cycles from the ice sheets coming down near the Canada-North American border at about 10 to 12,000 year cycles. And it just gives us a good way to track what's happening in the terrestrial vegetation system. And I'm sure this isn't a new figure to most of us in here, but this is CO2 data from Mauna Loa out in the monitoring site in Hawaii. And so on the left is annual means from 1960 up until this was 2024. And then on the right is that monthly mean, so we can see the seasonal shifts, summer versus winter CO2 levels. But it sets up where we're at right now with atmospheric composition and what we might expect to see in the terrestrial vegetation ecosystem and how it's responding. So my proxy, I said it just previously, is stomatal density, but that's defined as the number of stomata visible in the area that you're looking at, whether it's a leaf or a needle. And so we can infer significant signals from this metric, such as paleo CO2 with Van de Water, but in more recent years, with more robust methods, we're finding we can also infer paleo temperature. And what I'll be looking at today is stomatal conductance, and more specifically, the plasticity of stomatal conductance and just understanding how it shifts. So up in box A here, we have a Silurian fossil stomata that's from about 400 million years ago. So when they first evolved, we can see it in the fossil record. And it's diversified through time. So in box E, we have Pinus coreensis, or Korean pine. And I just want to note the difference between box G, which is a Poaceae species. Both form in stomatal rows, but in pines, we see that more pronounced stomata, whereas in grasses, we see that indented stomata. Oh, and just for fun, box B is a moss, and it looks really cool. So here's some information from all the papers that I've read over this past year. So up on that top left is a paper, Allworth et al. 2022. That's just showing the amount of CO2 that was in the atmosphere measured at micron moles at the time of growth for various species. So the main point that I want to make here is that we're seeing lots of clustering of the same characters. So plants are evolving with the same stomatal density relationship and similar CO2 levels. Down below that is Franks and Burling, 2009, where I'm getting my stomatal conductance calculations from. And so we're seeing, rather than that negative inverse trend of just stomatal density and CO2, we're seeing more of a positive trend with density and conductance. And this figure from Forelli on the right just helps visualize diffusivity time when we're looking at the size density relationship. So if we have high clustering, smaller size, the CO2 isn't going to travel through the pores as quickly as if it was a small size stomata with high density, similar to that top figure on the top box on the left. I kind of like to think of it like people trying to get through the subway. If you have all of the little gates open, everybody's going to flow through a lot quicker. But if several of them are out of order, there's going to be lines out the door. So Why did we even choose Great Base and bristlecone pine? Originally, when this work began and I was just tracking CO2, I was using huge geologic timescales from about 13.5 to 40,000 years ago. And I was using pack rat middens, very similar to the Vandewater paper from Ladder Cave out in Nevada. And it's an extremely long living tree. Methuselah out in Great Basin National Park is dated to be over 5,000 years old. I have a picture in a later slide of a tree we've collected from, we think is upwards of 2,000 years old. So it's showing extreme resiliency through climatic variability, through those glacial, interglacial cycles. And it generally tends, except for our hybridized bristlecones, it tends to be a very high elevation tree. So we're expecting to see more of a mixed signal of CO2 rather than signals from urban or regional hotspots. And that picture from Barber et al. Just shows very clear annual rings of the needles, which really helps us out in dating the fresh plant material that we do get. The pollen cones split them up very well. And they also can retain their needles for a very long time. As Bill was saying, we try to collect branches that have needles going back as far as possible. In a happy tree, that might be 12 to 20 years. But up in the White Mountains in California, where bristlecone are growing at over 10,000 feet, records from the Forest Service and the Park Service show it can retain needles for upwards of 50 to 60 years. So it's really holding on to those if it needs it, but if it doesn't, it'll shed them. Oh, and that little circle is Lake Canyon, where we just collected, so on that cusp of the range. And so coming into this study with the plasticity of stomatal conductance, I'm assuming that I can see measurable differences in stomatal conductance through changing atmospheric conditions. And I'm really hoping that I can use it on historical timescales. So we can track that Keeling curve, that hockey stick from the Mauna Loa, and really understand if the trees are reacting on this timescale to this level of change. So this is that tree I was talking about. This is up at the top of Lake Canyon after that grueling hike. And we've collected from these trees. So going into my methodology and my results, I'm going to narrow down my sample, my subsample to just the Garrett herbarium samples for a little bit of ease. And we're still processing the samples from the Stanley Welsh herbarium as well as Lake Canyon. It's another view of Lake Canyon Lake right there. But we're seeing a wide distribution of our points throughout all of Utah. And for my methodology, as I've stated previously, we've collected, we use herbarium records, and we use those plant macrofossils from Ladder Cave. And we went through quite a process of trying to find a good way to image them. But we found that using a cut down paintbrush to scrape that epicuticular wax and a microfiber cloth just to wipe it away leaves us with a good image of the stomata. We've tried boiling and zinc oxide, but it didn't work. And I'm using a 65mm lens at 5 times magnification and taking 50 pictures at 50 micron intervals to get a very high resolution image of that needle. I have a bigger one on my next slide. But I don't want to get too bogged down with all the software. So if anybody has any questions about it after, I'm happy to answer how we count and get the area of the stomata in more depth. So just before I show you the results, I just want to bring up some challenges to this study, especially with the Lake Canyon samples. We're really trying to understand, can the plant adapt in this short of a time scale, especially as such a long growing species and long living species. So it's something we're working through. And there have been a lot of assumptions on previous studies just because bristlecone pine is such an understudied species, let alone conifers in stomatal analyses. So we're really working with not a lot to go on. And the geometry of the bristlecone needles, it's a 5-needle pine, so that just makes it inherently more difficult than Edulis or Monophylla to image and get a good view of the stomates. Here's that zoomed in picture of the stomates. Pretty visible, and they definitely become visible after hours of staring at them. So this is that Google Earth workbook that I use to count the stomata. Each red dot is a stomata along the path function. Then I take the area of where I'm counting as well to get my stomatal density. And this is what we're seeing with stomatal conductance. So I know the R-squared value is very poor, but I don't want to discredit this work. We're seeing generally the same and preliminarily the same trend. And the red line is a linear regression and the green line is a lowest regression. Chose to use lowest because in finality, we will have multiple study sites and sample areas. And so we're really going to be trying to filter out the noise in this data. But we are seeing generally the same trend. Where it really gets interesting, though, is with the stomata density. We knew that this was the most reliable proxy going in, so it's not surprising that this is where most of the data or the results are promising. So this is stomatal density plotted against atmospheric CO2, similar to the Van de Water paper, and we're seeing that preliminary general negative trend inverse relationship. So it's exciting. This is probably my favorite graph that I've made. It's the size density relationship. So we're looking at the area of the stomates as well as the density, the frequency of how much they occur. And I think it's interesting. We're seeing some clustering here in this data. But compared to this graph here from Haworth, I think what we really need is more species, because what we're looking at here is a pocket or a threshold for Pilo. We only have one species and we're trying to understand how it's changing through time, but we don't have any other species to compare it to like Haworth does here. He's able to see the clustering and the patterns within certain species and how they're reacting to CO2 and how conductance is changing, or density is changing, excuse me. But I really do believe that even though we don't have robust results quite yet, it does not discredit this work, and it just pushes us in further directions for future research. And what does that look like? So as I stated, we need more species, whether that's different conifers or deciduous plants. We think that deciduous plants might give us a better signal of seasonality, a shorter response time to CO2, so we might be able to pick up higher plasticity or more changes, or simply more conifers with easier geometry, like Pinus monophylla, Pinus edulis, or Abies concolor. And new, really novel studies are coming out with genetic modification, targeting stomatal conductance for higher water use efficiencies. So I think this could be a really incredible and valuable methodology for resilient crops. And also going back to just that fossilized stomata 400 million years ago, we're in an era of abrupt climate change, and it's a problem that we are facing, and we need to go back and look at other eras that are similar, like the Paleocene, Eocene thermal maximum, and understanding, hey, what might happen in an era where it's unprecedented. So thank you, especially to the Garrett Herbarium and the Natural History Museum for empowering me to do this work and letting me use the collections as well as the Stanley Welsh Herbarium down at BYU. And to all the people on campus who have funded and helped out with this research, appreciate you so much. So thank you for listening, guys. These are all my references.
Thanks for questions. Okay, we have time for a few questions. I answered them all at the end, I think. Okay, if that's it, then that's it. Thank you very much. Great. All right. Our next presentation is by Alexi Olson and Dr. Tara Bishop from Utah Valley University. And they will be talking about ages and stages for juniper tree communities. So that should also